In this video, we'll study about the cell structure. Now, all living organisms, they are made up of cells. Some are made up of single cell and are called as unicellular organisms, and some are composed of multiple cells and they are called as multicellular organisms. So, the unicellular organisms, they are capable of independent existence and they are able to perform many functions which are required to carry out the life processes. So, that is why we can say that the cell is the structural and functional unit of life. Now, the cells are classified into the two main types. The first one is the prokaryotic cell and the second one is the eukaryotic cell. Now, the basic difference between the prokaryotic and the eukaryotic cell is of the nucleus. In case of prokaryotic cell, the nucleus is not surrounded by the nuclear envelope. The DNA lies naked in the cytoplasm of the cell, but in the case of eukaryotic cell, the DNA is surrounded by the nuclear envelope. The second difference is of the membrane bound organelles. In case of prokaryotic cell, the organelles which are present are not membrane bound, means they are membraneless, but in case of eukaryotic cell, all the organelles, they are surrounded by a unit membrane. Now, some of the examples of prokaryotic cells are bacteria. The second one is mycoplasma. Then we have blue-green algae. In case of eukaryotic cell, some of the examples are plant cells. Then we have animal cell. Then protists and fungi. Now, in this video, we will focus on the structure of the prokaryotic cell and we will take the bacterial cell as an example. Now, basically the bacteria are unicellular organisms. They are made up of single cell and they are devoid of chlorophyll. They have a naked DNA which is not surrounded by a nuclear envelope. It lies directly in the cytoplasm of the cell and they have the reserve food materials in the form of glycogen. Now, coming to the structure of the bacterial cell or a prokaryotic cell, there are following components present in the bacterial or a prokaryotic cell. The first one is the glycocalyx, then the cell wall, the plasma membrane, the cytoplasm and cytoplasmic inclusions, the nucleoid or the bacterial chromosome and the plasmids. Now, the first three components, they together form a structure, three-layer structure called as a cell envelope. Now, if you look at the diagram here, now this is the mucilage. We can, this is another name for glycocalyx. So, mucilage is the outermost coat of the bacterial cell and it is formed of polysaccharides, and in some bacteria it can also contain polypeptides. Glycocalyx is not essential for the survival of the bacterial cell, but it performs some of the very important functions in the bacterial cell. Like the first one, it protects the bacteria from harsh environmental conditions. Then it protects the bacteria from toxins present in the surroundings. Then it protects the bacteria from from viruses getting attached to it. Now the glycocalyx, if it is in the form of a layer which can be easily deformed and removed, then it is called as a slime layer. The second one, if the glycocalyx is rigid and tough, then it is called as the capsule. Now, the second one is the cell wall. It lies inner to the glycocalyx. Here, this is the cell wall. Now, the cell wall, it is formed of peptidoglycan. Now, peptidoglycan, this layer, it is a polymer which is formed of N-acetyl glucosamine and N-acetylmuramic acid and some polypeptides. Now, based on this peptidoglycan layer, the bacteria can be classified into the gram-positive bacteria and gram-negative bacteria. Now, in gram-positive bacteria, there are multiple layers of peptidoglycan, and in gram-negative bacteria, there is only a single layer of peptidoglycan. Now cell wall they provide protection to the cell and they also provide a particular shape to the cell. Now the next one is the plasma membrane. It lies inner to the cell wall. Here we can see the plasma membrane. Now the plasma membrane, they are formed of phospholipids, proteins and some polysaccharides, but they lack sterols. Now there is an exception that mycoplasma, they contain sterols. Now the plasma membrane, they allow certain substances to enter and exit the cell. It is basically semi-permeable in nature, it is thin and elastic. Now there are many structures which are associated with the plasma membrane. The first one is the mesosomes, the second one is the flagella, and then we have pili or fimbriae. Now mesosomes, here we can see in the diagram, these are the mesosomes. They are basically the infoldings of the plasma membrane. Mesosome perform some of the very important functions in the bacterial cell. The first function is that the mesosome, they help in cell respiration. Then the mesosome also help in cell wall formation. And the third one is that mesosome help in DNA replication and then the distribution of this duplicated DNA into the daughter organisms. Now the second structure is the flagella. Flagella, they are formed of a protein called as flagellin. So flagella, it is formed of a protein called as flagellin. Now the function of the flagella is that it provides motility to the bacteria. Some bacteria are motile and some are non-motile. Now the flagella, it is formed of three parts. The first one is the filament, the second one is the hook, and the third one is the basal body. Now as you can see, the filament, this lies external to the cell surface. The hook, it is anchored to the basal body and it is with the help of hook that it provides motion to the flagellum. Now the third one is the pili or fimbriae. Now you can see this is pili or fimbriae. They are thread-like structures formed, present all around the bacterial cell. These are the pili. The pili, they are formed of protein called pilins, and the pili they help in conjugation and they also help in cell attachment. Now the next one is the cytoplasm and cytoplasmic inclusions. The cytoplasm, as we can see here, they are granular. The cytoplasm is formed of 70S ribosomes, and here we can see which help in protein synthesis. Now here we can see the structure which is called as the polyribosomes. In this, the mRNA to mRNA, the multiple ribosomes are attached to form a structure called as polyribosomes. They help in the translation of mRNA into proteins. Now the next we can see is the cytoplasmic inclusions. Now cytoplasmic inclusions, they are non-structural, non-living component present in the bacterial cell. They are reserve materials and the examples of them are glycogen, the lipid globule, then we have synovial granules and also phosphates. Now the other one, if you see, is the nucleoid or the bacterial chromosome. Now if you see in the diagram, this is the bacterial chromosomes. Now this bacterial chromosome, it is circular double-stranded DNA molecule. See, if you see here, it has no free ends. It is basically the two ends are attached, covalently attached. It lies naked in the cytoplasm of the bacterial cell. And it is also called, the nucleoid is also called as genophore. Now the last one, the last structures present in the bacterial cell are the plasmids. Now they are not shown here in this diagram. Plasmids, basically they are not present in all the bacteria. They are present in some bacteria, for example E. coli. Plasmids, they are self-replicating extra chromosomal rings of the DNA. They provide certain unique characteristics features, phenotypic features to the bacteria, like they have genes for drug resistance, fertility factors, and they also have nitrogen fixing genes. Nowadays the plasmids, they are very important tools in the field of biotechnology and genetic engineering. That's all from the structure of the prokaryotic cell. Thank you.